I'm Tyson, and I've got a riddle for you. What do oxygen, potato chips, and the tires on my car all have in common? Any idea? They all come from plants. No kidding. Plants give us tons of stuff we rely on every day. Don't believe it? Okay. Say you took away everything in this room that came from a plant. Ready? Hold on one second. And go. I was playing that. Granny's table. Cotton. I mean, plants do just about everything, and they can do even more when we understand them more. Right now, scientists are cracking the code of life. Soon they will find secrets of plant genomes revealed.
Genomics has really changed the way we do biological research, and today we can access information we couldn't even dream about 10 years ago. And the experiments we're doing today really have the potential to change the world.
Right now, scientists everywhere are trying to make complete maps of the genomes of plants so we can understand how they got to be the way they are and how we can make better use of them in the future. But what the heck is a genome? Basically, a genome is the complete genetic information of any living thing. The tiny blueprint that tells a plant, a person, my pet goldfish, how to grow, when to grow, everything it needs to live. It's found in the nucleus of pretty much every cell, in that long ladder-like molecule called DNA. The rungs of the ladder are called base pairs, and a genome has hundreds of millions, even billions of rungs. Think of it this way. A genome is kind of like a big book, or better yet, an instruction manual. So, if one base pair of DNA is a word, the gene would be a paragraph, or one instruction like, make a root. Here's how. The genes are grouped inside chromosomes like chapters, and the whole book is the genome. But keep in mind, this book has as many words in it as are in this whole library. And, well, it's kind of a weird book. Because parts of it don't stay the same. Every generation, some of the genes change. They shuffle. So every little seed that sprouts gets a slightly different hand. And depending on the carbs, that plant might grow taller, smell sweeter, and if it's really lucky, it might even fight off a new disease. Now, wouldn't it be cool if we could peek at this hand? If we could read a plant's genetic code and help it do its plant thing better? Well, we can. We can make a map of its genome to find our way around it, and use that map to breed better plants. It's a process that involves gene sequencing, and it's going on right now.
My name is Elaine Martis. I work at the Genome Sequencing Center at Washington University School of Medicine. Sequencing is really like solving a big jigsaw puzzle.
Elaine and her crew have to identify each and every one of those millions or billions of base pairs in a plant's DNA.
It takes state-of-the-art computers, DNA sequencers, and robotic instrumentation.
With robots and computers running 24/7, it still takes years to sequence and map a plant's genome. But Elaine says they're getting better and faster.
It's always a challenge. There's always something new. There's always a new technology to wrap your mind around and start to think about.
Now, with billions of bits of information, it's up to experts like Michael Gonzalez to make sense of it all in an exploding field called bioinformatics.
This is the future of biology right here. So genes don't just raise their hand and say, hey, here I am. It takes intelligent systems to tell us where they begin and where they end. We can find genes, where those genes are located on a chromosome, as well as what those genes may be doing.
So we have a way to map all that DNA down to the last base pair. But what plant to map first? My friend Montona has the answer, and it's right under your feet.
Hi, I'm Montona, and I'm a Genome Project Manager. See this weed? This is Arabidopsis. It grows out of sidewalk cracks, in parking lots, it's everywhere. But this scruffy little thing has made history. It's the first plant to have its genome completely sequenced and mapped. It took scientists from 20 different countries working for 10 years to figure it all out. But now that we have a roadmap of this one plant, we can use it to compare the genomes of more complex organisms. There is a gene discovered in Arabidopsis. Someone found that we, humans, have the same gene. It's used to control our day-night cycles. Doesn't that just blow your mind?
Most definitely, my mind is blown. But just stick around, because we're going to meet people who right now are unlocking the secrets of three of the most important plants in our lives: corn, cotton, and potatoes. Let's go.
You have reached your destination.
I like cows. And they like me. They go, move. Hey, move over.
Out here in Middle America, corn is king. And we use it for tons of stuff. Cosmetics, latex paints, ethanol, crayons, disposable diapers, aspirin, shoe polish. I can't name everything. Can we run a crawl here? Thanks. Corn is an all-American plant, and we've grown it since day one. But did corn always look like this? No. A few thousand years ago, you wouldn't even recognize it. An ear of corn, if you could call it that, was about the size of my thumb. So how did it come to look like this? It's a genetic detective story. And here's the guy who tracked down the answer.
Hi, I'm John Cobley. I'm a genetics researcher at the University of Wisconsin, and I study the history of maize, what you call corn.
Using genomic techniques, John tracked corn's genetic clues to a grass-like plant from Mexico called Teosinte, which means grain of the gods. Supposedly corn's great-great-great-great-great-grandparents. Did ancient Native Americans really create the corn we know and love? I mean, come on! This is Teosinte, and this does not look like an ear of corn.
We've gone from something that had only 8 or 12 kernels in its ear to corn in modern times, which can have over 500 kernels in its ear.
Now, there's nothing better than sinking your teeth into those sweet, soft kernels of corn. But Teosinte's kernels are covered in cases as hard as rocks. What caused the change? By crossing Teosinte with corn, John was able to compare the genomes of the two plants, and he found his answer.
This plant has in it one gene that we've brought in from Teosinte that affects the structure of the ear. And if we pull off the husk, it shows you the dramatic effect that a single gene can have on the appearance of the plant.
Thanks to one gene, Teosinte's ears were turned inside out. Its hard outer cases became corn's inner cob, with the kernels now on the outside.
So basically, by selecting and breeding only for the things they wanted, the Native Americans turned a grass into corn. But it took hundreds, maybe even thousands of years. By understanding the genome, we could improve corn even more. Maybe make it drought-resistant or more nutritious. And it would take just a few years instead of 1,000.
We'd be doing the same thing the ancients did, only more efficiently and precisely. Because we'd know which of corn's 50,000 genes control the qualities we want. And the way to find out what a specific gene does? You knock it out. Turn it on, turn it off, and see what happens. You create a mutant.
Somewhere in this cornfield, there are mutants. Maybe a whole army of mutants. And inside their tall, leafy plant bodies are the mutant genes that make them look weird. And we will find them. Oh yes, we will.
We're in Berkeley, California, where driven, dedicated scientists are making mutants. The director of the Plant Gene Expression Center at Berkeley is Sarah Hake, who some call the Corn Lady.
I like the leaves, the tassel, the ear. I study the stem. I study the way it grows and the way it reproduces. I also dance like a corn dog.
Hit it!
Do the corn dog dance.
Out here we have mutants waiting to be found, waiting to be discovered.
So what we have here is a sexually confused corn plant. Normally, the male parts would grow up here, but as you can see, all the female parts are growing in the wrong place.
I'd like to show you one of my mutants that I'm working with, and what's interesting about it is that the leaf thinks is a different part of the plant, something that's completely abnormal.
So why are we so interested in mutants? Well, mutants help us figure out what the genes are actually doing in the corn plants.
Mutants provide a way to help map and study the function of each gene needed to build the different parts of a corn plant.
Our real goal at the end of the day is to know exactly what those genes are doing.
Sarah and her colleagues know genomics can make better corn plants. Corn that can provide better nutrition, that needs less water and fertilizer, that can produce bio-based fuels and products, improving our lives and the environment.
But they're just getting started. The ultimate goal is to figure out what each and every one of those 50,000 corn genes does. And the tools to reach that goal are improving every day. Okay, my favorite plant has to be cotton. I live in blue jeans. Cotton clothes are the best. Cotton's got seeds that grow fibers that can be spun into the softest threads. And when you put them on, you just got to move. Coming at it.
Can you cut into this? Can you cut into that? Yo. Can you cut into this? Can you cut into that?
Okay, let's bring it down. We're going to take you from shirt to dirt. From cotton jeans to cotton jeans. Ready? And go!
Rewind those blue genes all the way back to where plant cells are born, and you'll find yourself in a small cluster of cells called a meristem. Here cells get their genetic instructions for what part of the plant they'll become, be it stem, root, or fluffy cotton fiber. But just how did we get the cotton we know today? That's the question they're trying to answer at Iowa State University.
I'm Jonathan Wendell, and I study cotton here in Iowa, embedded in this ocean of soybeans and corn. Where's the cotton? It's up there. Come with me. Here we are in the greenhouses on top of the building. There's 1,000 species of plants up here, 50 species of cotton. The entire diversity of the world's cotton is here in this greenhouse. So here's the cotton fruit. And inside the fruit, like in all fruits, there's these seeds. And these seeds have attached to them all the cotton fiber. These cotton fibers are single cells. Look at that. Remarkable. Some of the longest single cells in the plant kingdom. You can take your fingers and you can pull out these single cells. Look at that. That one's 2 inches long. And they form these flattened spiral-like ribbons that'll stick together and can be spun into yarns and cloth.
By comparing the genomes of different cotton species, Jonathan and his team have pieced together the story of cotton's evolution.
Here's the plant that makes 90% of the world's cotton, of all of our clothing, of the blue jeans and the t-shirts you're wearing. This is Gossypium hirsutum.
So where did it come from? 10 million years ago, scraggly cotton ancestors evolved separately on different continents.
So here's Mom. Mom is from Africa. Here's Dad. Dad is from Mexico. How did they manage to get together? How can you interbreed when you're in different continents? Well, Mom crossed the ocean. We don't know how. Floating on a natural raft, maybe on a bit of volcanic pumice. Who knows? Maybe a constipated duck got blown off course in a storm.
Mom and Dad got together and a whole new cotton was born. A million years later, humans got hold of cotton, and through generations of plant breeding, made new and different kinds.
Humans turned something like this into this about 5,000 years ago through the domestication process. What we want to know is, what genes are different between this and this? That's what we're trying to learn in the lab.
So one of the things that we're studying in this lab is the fact that one species of cotton has 3.5 times the amount of DNA that several other species have, and we're not sure exactly why.
Computers to the rescue! Using bioinformatics programs, these guys can find patterns in the data. Patterns that might give us clues about how genes from different parents work together.
what isn't cool about cotton. I mean, you start off with these nasty little seeds, have these little itty-teeny fibers on them, and through domestication, evolution, we get these beautiful seeds with these long fibers that everybody's wearing.
Cotton is just so important. I mean, we'd all be running around naked, or at least in polyester, if it wasn't for cotton.
One day in the not-too-distant future, genome scientists will know which cotton genes do what, and their position and sequence in the genome. Then there won't be any need for guesswork. Breeders will be able to precisely track or move those genes as they breed a better plant. Like ones that grow longer fibers that make a pair of cotton jammies even softer than these. It won't be a dream. Hey, you guys want to turn off the lights when you're done?
When you got into this, when you got into that.
In the Andes Mountains of Peru, the potato plant first evolved. Cultivated by the Inca, taken by the Spanish, their delicious tubers spread far and wide.
Potatoes are a huge staple crop here and around the world. They grow where few things will grow. But imagine a world without potatoes. What if all the plants got sick and died? Well, it happened in Ireland, and it was deadly.
In Ireland, potatoes grew so well that the people became entirely dependent on them. Then, in 1845, a deadly disease called late potato blight wiped out Ireland's entire potato crop. It was called the Irish Potato Famine, and over 1 million people starved to death. Then, late blight mysteriously disappeared. But now the blight is back, and it's nastier than ever. Once infected, a healthy potato field quickly turns from vibrant and green to black with death. Here to inspect the devastation is Jim Bradeen from the University of Minnesota.
So late blight's really done its thing in this field. You can tell that it's a truly devastating disease. The first thing that happened was this spore germinated and grew into the plant. And once it's inside, it'll reproduce and will actually produce more spores that'll spread from plant to plant. And it can wipe out a potato field in as little as 7 to 10 days.
At genome research facilities, biologists race to find a genetic defense against this deadly disease.
I'm Barbara Baker, and these are my graduate students, and we're suiting up to go visit our potato plants. The suits are to protect the plants, not us. Late blight is everywhere, and we don't want to introduce it into this house.
In our research, we have lots of questions, like how does blight infect the plants?
So how do potato plants try and resist?
What genes are involved in putting up a fight?
The answers to all these questions are found in the potato genome.
Let me show you this. Spores of the potato late blight. You can see the lemon-shaped casings, and when water gets put on, you'll see the spores coming out.
This potato is susceptible to blight, but in nature, its ancestors are resistant. We want to understand that.
Potatoes and late blight evolved side by side, each making genetic changes to get the upper hand. Every time the plant develops a way to resist, the blight creates a new way to infect the plant. But now late blight has become even more devious.
Welcome to Club Spud. You want inside? I gotta see some ID.
The potato plants can fend off many kinds of blight because they recognize the blight's genetic makeup.
You, come here.
Huh? But some strains have a gene that potatoes don't recognize.
Oh, I know you guys.
No, we don't think so.
And those slip by like they're invisible.
Yeah, okay, you're in.
Woohoo! Thanks, man.
They walk right in and start an infection.
Okay.
Clearly, we need new solutions to the problem. We're very hopeful that genomics will help us get there.
In 1994, the unthinkable happened. A new strain of late blight devastated American potato crops across the northern plains. At the University of Wisconsin, the test plots of research scientist John Helgeson were no different.
Yes, this is the location where the late blight came through in 1994 with a devastating effect. We were looking at the fields around the 1st of August 1994. They were beautiful and green. 2 weeks later, it was completely leveled. It was just extreme. extremely dramatic.
But all was not lost. Amidst all the devastation, John saw reason for hope.
We saw a few green plants, green island sorts of plants here, and here that were obviously resistant.
The resistant plants came from a wild Mexican species, and somewhere in its genome was the gene that could fight the blight. The university hired geneticist Jimin Jiang, who worked with John to hunt down the blight-resistant gene. It took the team 4 years to find the gene, and another 3 years to breed a resistant plant ready for testing.
I was not completely confident because the inoculation test was done in greenhouse, which could be different from a true test in the field.
When we saw it in the field, when you can walk out there and we saw those plants in the field and happy and green, it was an exciting moment.
But just how successful were they? Let's head back to our blighted potato field in Minnesota.
Here's a bright spot in this otherwise bleak field. This plant has the gene from John Helgeson and Jimin Jiang, and you can see that late blight hasn't really even touched this plant, while it's completely destroyed the one right next door. And these plants are really living testimony to the power of genomics to protect the world food crop.
Farmers in the U.S. typically spray fungicides to fight late blight 12 times a year, which is a lot. Farmers in warmer, developing countries spray twice as much. Not only can they not afford it, but it's bad for the environment. It's a huge problem. A blight-resistant potato could eliminate thousands, even millions of tons of toxic chemicals from farming. So, fight the blight.
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 There's so many things that we can do with genomics once we know where genes are located.
Once we have the map, then we can ask so many more exciting questions about how all these things work together.
We can use that information to make changes and to improve on things we're interested in. For example, in agriculture, we can address issues in our environment. And of course, it's important for human health.
The research into plant genomes is already revolutionizing the field of biology.
We've seen how dynamic plant genomes are, and that it can take as little as one gene to change the way a plant looks and functions.
Genome research is increasing our understanding of nature, and it's giving us the tools to develop and breed new varieties of plants. It's really easy to forget all that plants do for us, the many ways they touch our lives, but they can do even more.
So we need to know how plants work, so they can work better for us. I tell you what, you can't beat the view.